Hello and welcome to Chapter 9, Infection Control Concepts. In this chapter, we will take a look at microorganisms in addition to some viral diseases, the cycle infection, some types of portals for exit and pathogenic organisms, and ways that these can be transmitted throughout the hospital. When we start to look at microorganisms, the hospital is a great place for gathering lots of diseases and lots of sicknesses, and it's a route of transmission that can occur that you may not be aware of, or you stop to think about how things can be transmitted. So with microorganisms, there are living organisms that are too small to be seen with your eye. They're going to include bacteria, viruses, protozoa, prions, and fungi. With bacteria, some of the things that you should be aware of is that they are a small single-celled organism with a cell wall and an atypical nucleus that lacks a membrane. The cell wall is essential for survival of bacterium, and it makes a target for destruction by some antibiotics. So that's the good news with these, is that they can be cured in most cases with antibiotics. The types of bacteria that you may see or may be familiar with are strep throat, bacterial pneumonia, and food poisoning. When we look at how they are classified, bacteria tend to be independently replaced with a host cell. Most bacteria have one or three distinct shapes. They're spherical, they may be spiral, and they may be rod-shaped. Cocci and bacilli may also be further classified based on their cell groups. So they could be singly in a group of two with long chains or clusters, and bacilli can occur in single cells in pairs and chains. Bacteria also requires oxygen to grow, and they're called obligate aerobes, which others will not grow in the presence of oxygen, which are called anaerobes. Bacteria can adapt and grow under aerobic and anaerobic conditions. When we take a look at viruses, viruses are a subcellular organism that are among the smallest known disease organisms. They're small in size, and they can be viewed with a microscope. They can be fully developed and called a viron, which is made up of genetic material, which consists of the DNA and RNA. So you need to have both DNA and RNA, never both. It's DNA or RNA. The capstone may be covered with a lipoprotein that involves spikes on the edges. Those enveloped viruses are seen in common colds, the flu, cold sores, warts, and in even the cases of mono. Fungi occurs in a single-celled yeast or long structure called mold, and they are composed of many cells. Some fungi will exist in either form depending on the environment. Yeast is going to reproduce by forming buds of molds, which call spore formation. There are more than 100,000 types of fungi in the world. Sometimes we do use fungi in things that we eat or drink, such as alcoholic beverages or flavored cheeses. But some fungi can be destructive to natural materials like wood and leather, and they can cause diseases in plants and in us. So some examples of fungi can take place in your hair, nails, or skin. And a lot of times you'll see patients come in with a type of athlete's foot. They may have ringworm, they may have some respiratory infections that occur from inhaling the spores. The last one we're going to take a look at is prion. These are the smallest and least understood of pathogens. They were discovered in 1983, so they're relatively new. They have an infectious protein. They do not consist of an RNA or DNA, and they are resistant to the body's natural defense. One of the things that, and we've been talking about in class, is dementia and Alzheimer's. So when we look at these, they tend to be a lead causer of dementia, and they do cause neurological changes in the brain. Protozoan parasites are unicellular. They generally exist in free-living organisms, and a few do have parasitic and live. They will live inside the body. They do not have a cell wall. They tend to be very resistant in form, so the example would be like cyst. So they can survive in an environment outside of the host. Some common diseases that you may see in this classification are sexually transmitted diseases and toxoplasmosis, which is contracted from cat feces and eating undercooked meat. There's an establishment of infectious diseases. So we do need to have a host. That infection disease must overcome the host and cause the disease, and the host and infection compromises and lives together, which leads us up to our cycle of infection. So the factors involved in the spread of the disease are sometimes called the cycle of infection, which can be transmitted through an infectious organism. When we look at an infectious organism, that microorganism is capable of causing a disease of pathogenic organism or pathogens that possess certain properties and factors that distinguish them from non-pathogenic organisms like normal flora. So when we look in here, in your textbook, they give you a lot of examples of those pathogens. I just noticed that this says table 8-1. This should say table 9-1, which is found on page 155 in your book. So please make a note of that mistake. On table 9.1 are examples of pathogens. Respiratory tract, we have our urinary tract, the skin, GI tract, your blood are all examples of areas in your body that could have common pathogens. The reservoir of infection is a place where pathogens can thrive in sufficient numbers to pose a threat. Some conditions that they must have will be moisture, nutrients, and a suitable temperature. So in a human, it makes an ideal location for these to thrive. We can also see in animals, salmonella, in soil that reserves for tetanus, food, and water are also areas that can contain and contaminate from these sources. You must have a portal of exit from the human body, so the route of blood, bodily fluids, excretions. So our GI tract, any open wounds or respiratory tract are ideal for this. And you must have a susceptible host. So your patients are often host because of their reduced immune system, because they're fighting some other type of disease while they're in the hospital. They are known to be very susceptible to catching these disease processes. So when we look at a host, we have what's called nosocomial infection, which is also called an HAI, which is healthcare associated infection. And some examples of those can be found in chapter 10 when we talk about those. But our healthcare workers are also at risk because of blood and contaminated bodily fluids. So it's very important for you to follow infection control processes to reduce your chances of having that disease process. So we're really concerned about our portal of entry, and so there are a lot of isolation techniques that we use to help reduce that. We don't want to have a microorganism that gets through the respiratory tract. If it's on your hands, it can get through your urinary tract, the GI tract, mucous membranes of your eyes, nose, or mouth, or bloodstream. So any of those breakdowns will cause you to allow that pathogen in. So infections that are acquired in your hospital are called nosocomial infections. They are affecting the patient, they affect you, and they are called occupationally acquired infections. I also want to remind you of that term normal floral. which can cause infections when they're located outside their usual environment or when the host immune system is compromised. So these nosocomial infections are the 8th leading cause of death in the United States. The most common one that you're going to see take place in the hospital system is a UTI, and generally that's because of Foley catheters. We need to be concerned as a radiographer because we could be susceptible host as well if we don't use proper isolation techniques and wash our hands. So those with compromised or immune suppressed we're very concerned about. That's why we have strict precautions when we enter those rooms. They have very weak immune systems, and so we need to protect them from catching something that we may be transmitting to them or other patients. So patients who are having chemotherapy, they may have HIV, organ transplants, open heart surgery. You will see notices placed on their door, and it will tell you what type of precautions are taking place before you even enter the room. I've given you an example of some signs that could be handed or hanging outside the door, but they do have other symbols and other types of signs that you need to be aware of for your institution. So some sources of those nosocomial infections: medical personnel such as ourselves, our patient, any contaminated hospital environment, so things that have been touched and not properly cleaned, bloodborne pathogens, and any invasive procedures where there may be an open wound or a route of entry. Those transmissions of disease can occur in different ways, so the most direct way to break the cycle of infection is to prevent that transmission and be aware of it. The 6 main routes of transmissions are direct contact, fomites, vectors, vehicles, airborne, and droplet contamination. When we take a look at direct contact, this is the first route. This transmission mode requires that the host is touched by an infected person and that the organisms are placed in direct contact with that tissue. Some examples of these are syphilis, HIV infections, and staph infections. So these are contracted when that infectious organism on that individual are placed in direct contact with the mucous membrane of the host. Skin infections can occur in the hospital workers because we have frequent contact with patients. So there are staph infection. They will have signs for precautions in those rooms, and they will let you know that this person has something like MRSA. A fomite is an object that's been in contact with a pathogenic organism. These could be urinary catheters. It could be a contaminated glove, an x-ray table, positioning sponges, anything that has any bodily fluids that were not properly treated. A vector is when a body or infectious organism develops multiples before becoming infected to the new host. Bites by infected insects can transmit diseases to humans. We often see this in mosquitoes and ticks. So a vector, even bats, can transmit rabies. And so those are things that you need to be aware of. So in your registry review content that's put out by the ASRT for getting you ready for your ARRT, 2 items are individually listed on those: fomites versus vectors. So what I want you to remember about a fomite is, again, this is something that's been contaminated. Think about those cruise ships where they become severely infected by a stomach virus, and then everybody's put on quarantine. What typically happens is they go and they clean those rooms with a mop or a cloth. They don't properly disinfect those objects before moving on to the next room. So if you have a mop water that's been contaminated and you're going and you're cleaning other rooms, you're transmitting that disease process from room to room. So a fomite could be something other than the urinary catheter. It could be something like a mop. A vector, again, is going to be something that has been infected by an insect bite or something like a bat. So those have to be transmitted through that bite. So we're not going to see a vector taking place with a radiographer. You may see it with your patient, but not you. A vehicle is a mode of transportation for that microorganism. We see this take place a lot in food, water, drugs, or blood. This is one of the things that we encourage you to get your hepatitis A vaccine. So you do have a B that's a required, but I do suggest getting your Hep A as well to help to avoid any contaminated food that you may come in contact with. Transmissions based on precautions. These precautions can be airborne, droplet, contact, or reverse. With contact procedures, this is anyone who's been touched by that individual. We do have MRSA that takes place. MRSA is something that naturally occurs in the human body, but too much of it is a bad thing. So you could have the flesh-eating disease is MRSA, and it can be cured right away. So if that individual goes to the emergency department and receives some very strong antibiotics when they first notice it on their skin, then they can go home that day. But when it becomes worse and starts to spread more, that's where we have to hospitalize them, and then contact precautions need to take place. Hep A, varicella are all examples of things that may require contact precautions. So with contact precautions, you want to make sure that you're wearing the proper gloves. You have your isolation gowns, which are usually yellow, but there are some institutions that have blue ones. And you do want to wash your hands. Washing your hands is the best way to protect yourself when you get out there. Surgical mask may be used if you're suspecting that there may be body fluids that are splashed up into your face. So droplet contamination often occurs in infection. Individual coughs, they sneeze, they speak, they may be singing in front of you. So those droplets are airborne. So this is any mucous membrane of the eyes, nose, or mouth from that host with large droplets that are greater than 5 micrometers that contain microorganisms. Those droplets do not travel very far. They're going to travel about 3 feet or less. So some examples are influenza that you would be familiar with. So your flu, meningitis, diphtheria, these are all things that you'll notice you had immunization vaccines for. So those droplet precautions, we do want to take into consideration that they do travel 3 feet. So usually when we can wear eye protection or a mask, we want to wash our hands, we want to have our gown to make sure that we don't take those disease processes home to our family. Airborne transmission occurs from dust that contains spores or droplet nuclei. So these droplet nuclei can travel up to 5 microns or micrometers or smaller. So these droplets are not very big, and they can remain suspended in the air for long periods of time. So with these, you'll see a lot of patients that if they have TB, they wear a mask. Also, varicella viruses can be transmitted direct contact. So those can be transmitted in the form on the skin of patients, and then they infect with that virus. Airborne precautions for these, because they do remain in the air for long periods of time, you may be required to be fitted at your hospitals for an N95 mask. So you'll wear that N95 mask or a face shield, and then you'll notice that the rooms are negative pressure rooms, so they are pumping in air, fresh air into those rooms to help combat that. So varicella and measles, we saw a measles outbreak take place about a couple of years ago on the West Coast. And these were your anti-vaxxers who did not have their kids vaccinated. So they had a strong measles population that pops up. With those folks, you're going to have to protect yourself so you're not carrying it to other people that may not be vaccinated in the hospital. So that's why they take precautions in place so that you're not spreading that throughout the hospital. Again, hand washing is the best thing you can do. Wash your hands before you go in with a patient. Wash them when you come out of the patient's room. When you go into these rooms, you may be required to wear your isolation gown. You may have to wear a cap, a mask, and gloves. Our X-ray plates, so if you have a DR imaging plate, you do want to place a plastic cover over that plate so that when it comes in contact with your patient, it's touching the plastic, and then you can throw the plastic away. So here's an example of them putting that plastic on the plate, and here's an example of them disgowning and taking off the plastic from that plate before they exit the room. So your human body defenses are your skin, mucous membranes, your sweat glands. These are all points of entry that can occur. And they can occur in everyday activities that you don't think about. So even when we're out grocery shopping. So again, the best route of defense for you is going to be washing your hand. We do have some natural resistance that we are born with. And we may have immunization or vaccines. So there is some environmental control that can take place. So medical asepsis is when we try to reduce the infectious agents in that hospital environment. We can do sterilization of our surgical instruments and kill out those agents after surgery. Our temperature also plays a role, so we do want to keep rooms at a constant temperature, and that pH factor is also a factor. We have chemical methods that we can disinfect. We can use antiseptic disinfectant, which is a topical disinfectant. Those gas sterilization chambers will actually kill off organisms by heat. And the effectiveness of those are going to be your chemical concentration, temperature, time exposure, and the nature of the person being treated and following those isolation techniques. Here's an example of what an autoclave looks like. With an autoclave, we do set this up. We put the instruments, so the surgical instruments in here. You'll see that it can, under a moist autoclave, that steam works. And it actually heats up to about 250 degrees Fahrenheit for 15 minutes. Then we have a dry oven that has a higher temperature for longer periods of time, which is a 320 degree Fahrenheit for 120 minutes. And what that does is it just slowly cooks off those microorganisms. You can do a pasteurization, which is a moderate heat and rapid cooling, freezing, and UV light as well. But in the hospital system, especially if you're working at an urgent care and you're doing a role of a medical assistant and a tech, you will see that you may be required to run that autoclave system. So that's something that you would get on the job. You'll learn how to wrap those instruments, place them in the autoclave, set those controls, and most of them that you'll be using will be the moist ones. So they have that steam pressure. Hand washing, again, is your most effective way to prevent and not getting infected. So hand washing should be done, and most of you learn to the songs of like the ABCs. Sing the ABCs in your head. So make sure you're properly washing your hands so that you have a defense against infection. Thank you so much. And I hope you enjoyed this chapter on infection control concepts.